Okay, let's get started with today's session. Today is for Unit 6, which is about the SNP. And how's your group project going? Yeah, and actually group project is about the SNP. So today I'm going to introduce some basic information about the SNP. But when you actually do the group project, you want to research on a SNP which is not researched extensively, and to perform some data analysis, and then write the manuscript based on your hypothesis. So eventually, you either prove your hypothesis or disprove your hypothesis based on the data and results. So for your group project, this is going to be a fundamental basic knowledge. So so let's get started on that. So let's first start by the topic. So today we will focus on the SNP, single nucleotide polymorphism. Why are we interested? Because it may result in different phenotype, like a disease, for example. But is this? So I'm going to tell this in detail in the next few slides. But it's just SNP is a kind of a DNA variant compared to the reference genome. Most common variant, and yeah. However, that's not the only variant. There's a copy number variation, CNV, and that's not all. We have in more general, we have a structural variant. So SNP is a single base pair level, and then insertion and small insertion and deletion, and then copy number, and then structural variant. So it's going like that. So basically, we are concentrating on the difference between your sequence and the reference sequence. Because hypothetically, reference sequence is from the normal regular person. So the basic idea is we assume the reference sequence is normal and not problematic. When you have some DNA variant, now we investigate this in further detail and take a look at what effect does it cause. So it's kind of a difference between DNA sequence, and we will actually categorize and study step by step. So how to get this SNP data? There are many databases, OMIM, Biomart, UCSC, Galaxy, IGV, 1000 Genome Project, ClinVar, GenomeAd, and there are so many resources. Why are these matter? Because you guys are studying the SNPs for your own group project. Your own group project is pick a SNP or variant, and then study the functional role. How does it affect the phenotype? Yeah, so all these databases are actually relevant to your group project. So before we get into this into each technical detail, if I just provide some the objective or some more detail, you want to check OMIM database when you actually it's about the Mendelian like a heritable disease database in relation to the SNP or variant. So when you study on your SNP in relevance to the disease phenotype, yeah, you want to search the OMIM, and also you also check out the DB SNP database. If you haven't done it yet, yeah, I strongly recommend. check out the content and try to understand um, all the details in the dbSNP. And then you wanna find the all the so for example, your SNP is located in some gene, right? I hope that you choose the SNP in the genetic gene region, not like an intergene region. Intergene means between the gene, so that gene is not associated. It's possible that some SNP in the intergenic region is associated with your phenotype by like being an enhancer region or some like a super enhancer, and there are some other mechanisms of or non-coding. It's possible, but the low-hanging fruit is your variant is located in coding sequence of the gene and change the amino acid sequence. That is a low-hanging fruit. Yep. So yeah, you when you do the group project, I wanna you guys if you chose the SNP that is the coding region, yeah, that make a lot of things easier for you. Otherwise, you have to dig up much more, and direct association is not easy to get for intergenic or intronic, unless in the intronic case, it's located in the splice site region or non-coding region. And then you can get the you should be able to get the SNP information from Biomart and Galaxy, and you want to visualize those SNP information in the IGV. Last week we did we prepared the bed file of the SNP. I probably wrote here. So if I do the chromosome, any a SNP at 1 million position. It's an ending position of the interval, and if this becomes editable to the bed file, 0 bed, so that you can easily upload to the UCSC Genome browser. Instead of if you write down the same number here, the interval length does not span any one base pair, it's just line. So it's not, so UCSC or the IGV use the 0 bed. So the position here is the 1 million minus 1, 99. So if you remember this 0 bed file for the SNP, so actually the location of the SNP is this at this point. And this is just the to load the interval. So the last week we uploaded this bed file to the IGB and actually visualized, hey, here is a black interval in IGB. So that is the view the SNP in using IGB and navigate the 1000 genome browser or look up the 1000 genome database. And there are so many things that we can do. So and search the ClinVar, the flag SNP, which are in the UCSC database, and there are many other things that you can do with the SNP. So these actually give you ideas on if you just ask. So my group chose the one SNP, what to do? Yeah, there are a bunch of things that you can do, and this is not all actually. This is just the beginning of your research. Okay, now let's get started on the technical detail of this. SNP is a type of polymorphism. Polymorphic means many, right? Morphism means forms, right? So many forms. So for the DNA chromosome 1 ACGT sequence, right? And at a certain position, we have some variance in the population. Yeah, for example, let's show the hypothetical chromosome 1, the FASTA file, reference genome file, hypothetical one. So FASTA file, the sequence file, the FASTA always starts with a greater than sign, and then followed by a PHR, one, right? And then now I write down the ACGT. So hypothetically ACGT, right? So let's just say that hypothetically this is the reference genome of chromosome 1, and it goes like that for 250 million base pairs, right? So, and let's just say hypothetically assume that this is a human. So reference genome of a human sequence, and now we sequence another person, and then another person, chromosome 1, and the sequence, since we are human, it's mostly the same. 1 out of 1,000 base pairs are going to be different. So mostly the same. A, C, D, C, A, C, D, C, A. And however, for this C, let's say that this person has it. I just make it up. And then the rest is the same. So this C changed into T for that person. This for the reference, it's a C allele. And then for the it's a T allele. And can I say that this region, chromosome 1, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, chromosome 1, 11th position, is it polymorphic? Polymorphic means these kinds of incidents are found not so rarely, at least by 1% of the population. So based on 1%, we should not say that. But when we sequence about 1,000 people, at least the 10 people show T allele, or more than 10, so that, yeah, we can say that, hey, that is the polymorphic region. The chromosome 1 11th position is the polymorphic region. And let's study. In this case, the DNA sequence of the C is replaced, not inserted, not deleted, but replaced with a T, right? T sequence, the substitution. And that's the SNP, right? So SNP is the actually the easiest to study. And then it's most common DNA variant when you compare your sequence against the reference field. Any questions so far? It's pretty basic, right? Yeah.
You said that the polymorphism should occur in at least 1% of the population
In general.
in general. So, like, how is it quantified? Now, suppose, is there a standard which is said, like, okay, the sample size should at least be of 100 patients or 200 patients so that we can conclude it?
Yeah. So this is called allele frequency, right? Assuming 1,000 people is sequence, among 1,000 people, 900 have this C allele, and then 100 has a P allele. Then allele frequency is the 10%, right? So initially we started seeming to be not so much rare by defining the supporting mobility, so at least 1%. But as we study more and more, we found a more rare incidence. So even though the concept started, hey, it should be somewhat present in the population so that not so much rare, like a 1 out of million people have it. That's too rare. So let's just focus on at least some alleles which have at least 1% allele frequency. That's the starting. But nowadays, that is no longer true. We study many variants, which is less than 1%. So a lot of SNPs actual like allele frequency is now, if you look at the allele frequency table of a SNP, it often goes 10 to the minus 7 or 2. Okay. Any questions? All right. Now, I would like to do some group exercise. This is our main group exercise today. And I would like to spend some significant amount of time. And let's use our group, your group in the MS Team. Group 1, group 2, group 3, group 4. Everybody has access to MS Team, right? Yeah, or you can sit together because I'm going to give it. This exercise will take at least 10 or 20 minutes, or even 30 minutes, who knows. But I want you guys actually try before I discuss it. So, before we get started, let me give you some introduction. How many of you are familiar with the Alzheimer's disease? Everybody heard about it, right? Alzheimer's. Yeah. What caused the Alzheimer's disease? Do you happen to know it? Can you share your knowledge or view on the Alzheimer's disease? Since everybody knows about it. So what causes it? Pardon?
I believe that the exact cause is not found in. They believe that there are a lot of causes and epigenetic factors causing the disease. So we don't exactly know what particular causes.
Yeah, that's true. We don't know at this time what exactly causes the Alzheimer's disease. Actually, I actually got my PhD in the Alzheimer's disease research. I mean, look at I did a study. But yeah, even though I got the PhD for that, yeah, unfortunately, we don't know. So we don't know the exact cause of Alzheimer's disease at this time. That's bad. Second, when you become older than 85 years old, do you have to know the chance of getting the Alzheimer's? Yeah, but when you reach that 85-year-old, how much percent of the population in general have some symptoms related to Alzheimer's?
Maybe 60%.
Yeah, more than half. It's pretty. So it's not easy to avoid that. Third, drug. Is there any drug that cure Alzheimer's? No. Now, the second question is there are some FDA approved drugs that claim to be reduce the progression
of the IPM. Have we actually checked by how much or how much percent of people? Yeah, check it. It depends on, but I want to actually see that. And the actual effectiveness of the drug, not even curing, but what do you say? The slowing down the disease or making it better. Actually, it's pretty poor. You'll be surprised to see that kind of number. So, in summary, we don't know what it causes at this time. We don't have a drug to cure. We have some drugs to alleviate the symptoms, but it's not working that well. And then in recent 10 years, or 10 years, or 15 to 10 years, only very small number, probably 1 or 2, were newly approved by the FDA. And recently, one in about 1 or 2 years ago. And the effect of that is not clear. In my opinion, that drug should not be approved. And I actually saw the data. That's my personal view. But anyway, it's approved with a marginal effect. So that's the current situation. However, we know something. How many of you heard about APOE4? Please raise it. Yeah, this is the, even though we don't know the cause and we don't know all this, when you statistically study the person with APOE4 type has very high increased Alzheimer's disease risk. That's very sure. You cannot deny that when you actually get the data. So don't you want to know what's going on here? Yeah, that's basically the group, the class exercise. So the normal people has APOE3 type. But if you happen to have APOE4 type, actually what we call the A44 protein E, the epsilon 4 type, if you have that kind of a genotype, then the statistics clearly show that you have very high risk for getting the Alzheimer's. So you might want to ask what's going on here. So let's try to investigate that together. So APOE3 is a normal people's the genotype. APOE4 is the genotype of people with an increased Alzheimer's disease risk. Can you? Oh, I answered the Q1 then. All right. Yeah. Okay. Let's start from the question 2. Can you perform the DNA sequence alignment of these two APOE3 type and then APOE4 type? And then can you show something like this? This is not the APO3 versus APO4, but imagine the top one is APO3 and bottom one is APO4. Show which one is different by doing, so that's basically the SNP. So we want to actually find out the SNP in APOE4 type. Since APOE3 is the normal, so you guys want to find out which SNP is responsible for making a person to a POE4 type. In that case, you guys have that person has increased item with this
Yes, feel free to move around. Yeah, if you guys want me to stay out of the room, I can. Yeah. I will come back in like 5 minutes, and then feel free to speak each other and discuss freely and check whether you guys are done. So how many groups finished the number one, number two, to perform the DNA sequence alignment? Please raise your hand if you guys finished. No, one group, two groups. The other group, do you guys need some more time? Yeah, I would like to ask you, one of you actually volunteer to present so that the other people get to know how you guys did. And then if the other group did some different approach, please let us know and also present. Yeah, let's get started from the one group. Is there anyone who wanna volunteer and try to show how you guys do the sequence alignment of ApoE3 and ApoE4 DNA sequence? Yeah, please share your screen. And yeah, don't feel have too much pressure. If you're wrong or if you get stuck, I'm here to help you. So yeah, please share the screen and then let's go over together. There are so many ways doing this.
Says that we can share our screen.
You cannot? Oh, sorry. Oh, I have to. Yeah, can you share your screen now? All right. So let's get started from at the end first. Can you go around the 2/3 position, 70% of around here? Did 100% match? There should be one. And All right. How did you get the subject sequence and query sequence?
So I adjusted the activities rate.
Oh, you did a cloning vector. Okay. Okay. Cloning vector is not really the good idea because it may contain some other parts of it. So good try, but this is not really the representative sequence of the ApoE4. So that's why it's not that easy. Yeah, but basically, you find the subject sequence, you find the query sequence. You are comparing subject and query by using the blast. That's basically what I was trying to do. But the only problem was the subject and query sequence was not from standard ApoE3 and ApoE4 sequence, but the cloning vector sequence. So that was the origin of the problem. So how about the other group? Thank you for sharing. Yeah. The other group, can you actually share your screen and then let's discuss it together?
So we just got the pasta sequence, so I just run the blast. So with the protein sequence, we ran blast 10, blast 10, and from here if you see there is a transcript variant 3 for this one, and if you see here there's a transcript variant 4. So we're gonna take these sequences and we're gonna run blast for it.
Yeah.
This is the first one.
So where is the difference? I see. Okay. Good try, but this time you tried to get the transcript sequence and then try to align those transcript sequence. In principle, this is not a wrong approach, as usual. And is there any other group? Thank you for sharing it. Yeah. Is there any other group who did successfully? Yep, that's okay. Let me try. Let's try it together. There are so many ways. That's why I asked you to try it. I would like to Google ApoE4 UniProt database. It's a one database. It's just one way of doing it. Oh, yeah, thank you for telling me. Yeah. So let me Google ApoE4 UniProt. UniProt is a database. Yeah, can you guys do it together with me? So let's do it. And there is the Apo. Yeah, actually, I didn't do it. So I'm actually trying it right now. So I cannot 100% guarantee the result, but let's try it. Theoretically, this should work. But let's try it. So ApoE4, which is the variant allele, the SNP allele, which has a problematic part SNP in it. And this is apolipoprotein E isoform 4. Yeah, that's right. Let me go there. And then there is a sequence tab. Do you see the sequence tab from the left? Right? And here is the sequence, right? Good. So, all right. So I know what's my target, but what's my reference? My reference should be ApoE3. So let's Google ApoE3 UniProt. But is that the standard one? Just ApoE, right? ApoE3 type 3 is the regular one, so it should be the same as ApoE. So when I actually Google search it, yeah, it indeed show just regular apolipoprotein E, which is the ApoE3 type. So here is the IDE, and then yeah, right. And many people actually suggested the BLAST NCBI to sequence alignment. So can you search the BLAST NCBI? Right, I went there, and I want to do the nucleotide BLAST. So I click the nucleotide BLAST. I want to compare DNA sequence. Here, it's very important. I want to sequence only 2 sequences that I'm going to provide, not 1 sequence against whole database. But default mode of BLAST is you provide 1 query sequence, search against full of the data set. That's not what I want. I want to provide a 2 sequence, ApoE3 and ApoE4, and just align those 2. And do you see this check mark, align 2 or more sequences yourself? So I'm going to click that. So when I do that, I have a query and subject. It doesn't matter where to put which one. It as long as you know what you did. And ideally, let's try. Trying is actually not bad. So for the query, let me just put the UniProt ID, hoping that NCBI works. And I hope that it works. And the corresponding ID is this one, and yeah, if it doesn't work, don't blame. And it didn't work. Yeah. Okay. Yeah, obviously it didn't work. This is the amino acid sequence. That's actually the problem 3 that I wanted to do. All right, let's do that first. In that case, I want to do the BLAST N, protein BLAST, protein sequence to protein sequence. And yeah, let's try again. Did it work? So instead, I just I did the protein sequence here, and for the ApoE4 type, there is a blast link, and here is the that protein sequence, and let's do the blast. Right. Boom. Okay, just one moment. Let me put up something else. Okay, let me share my screen again. Yeah, in genomics, there are many ways to do this, but I would like to show this way. This is just one particular way. You guys actually try to get the sequence from NCBI database and do the blast search. That is the one way to do it, but there are many other ways. But I would like to show the way that I have done. So I'm going to share this the resource together with you. So this is the DALI, and I just share it into the chat. Right now, I gave a hint for APOE3 is 1 LPE. APOE4 is the 1 GS9, right? So in the DALI, this is, let me try to search in a pairwise. Click this a pairwise, right? And then the first one is the A1 LPE, but it is asking, do you want to have specify the chain? If it has a multiple chains, chain 1, chain 2, chain 3, chain B, chain like 4, chain 5 like that, I want to specify the first chain. First chain is always A, capital letter. So I put the capital A here. Now I'm going to press the plus button here. And the PDB ID for the APOE4 is the 1GS9, and again the chain A. So when you're ready, just submit. So again, this is one pairwise the database for APOE3 with the chain number A, APOE4 with the chain number A, and then submit. You will get something like do you want to get the HTML file? So click that HTML file. And you should see, interactive HTML. So if I click the interactive HTML, and this is the sequence alignment in a protein level, and cysteine to the arginine change in here. The rest are the same. Yeah. So in the query sequence of APOE3, cysteine is the cysteine, and then the subject sequence, I put the arginine, right? So that is the perform the structure. One way of perform the structural sequence alignment. DNA sequence alignment is about the same. You just get the that and blast it, or do some specialized database to do these kind of things, right? Now, have you ever heard the structural alignment? Yeah. What? Yeah.
So is PDB the only database that has, I guess, reliable information?
This is one of the many.
Okay.
Unit6, and then the NCBI ensemble, UCSD, anywhere you can go and then get that.
say the DNA sequence?
So I will leave the as a homework. Try to find the reliable A4E3 sequence and then reliable A4E4 sequence, and then do the alignment. And you should see some SNPs. One of them in the coding region, there is only one in the coding region. That is the one that changes arginine to the cysteine to arginine. So cysteine is C, arginine is R. So that's the one that you have seen, the protein sequence alignment. And this figure actually says the general public has 8.4% of ApoE2 type, 77% of E3 type, 13% of E4 type. But in Alzheimer's disease, the ApoE4 type has very high percentage, 36.7%. But the ApoE3, even though it's large compared to that, the odd ratio, it's relatively small. And this one is even smaller. So by looking at this allele frequency table, we think we claim E2 type is somewhat protective. E4 type is the AD causing type. So, but it's not black and white. It has it has some some like a percentage, right? Yeah.
So is the SNP just seen on the protein level and not nucleotide?
Oh no, there is you can actually do that in the DNA level.
So, but on the DNA level, there will be changing more than one basis, right?
Yeah, it's changing one basis because by looking at it, actually, if you look up the literature, you'll find that there should be 3, but only one in the coding region, which is this.
So while identifying SNPs, we just see one amino acid is changed, but then that can result in change in more than one nucleotide, right?
Theoretically, that's right. But when we actually talk about this in a canonical form, yeah, it's indeed only one variant. I will introduce that AS.410.635.01.FA23 that SNP. So we will talk about that in a few moments. But at this time, just think about this as DNA sequence-wise, there are multiple SNPs, 2 in the non-coding region, 1 in the coding region, and then the coding region 1 caused the amino acid change, cysteine to the arginine. Yeah, that's the message. Now, let's do the structural alignment. Sequence alignment is ACGT sequence alignment or amino acid sequence alignment. But structural alignment is different. So can you actually try this? So let me just write down the PDB ID here. One LPE, AOE3, and then the 1GS9 or AOE4 type, the problematic one, right? So let's try this together. Protein data bank. We are trying to do the structural alignment. Do you see this RCSB PDB homepage? Yeah, let's go there. And can you bring your mouse to the analyze here? Do you see pairwise structural alignment tool? The first one? And do that. And now just type the first one, 1LPE, chain A, 1GS9, use the default parameter, and click compare button. Did you do that? So let me click that. Do you see this structural alignment? It's almost perfectly aligned. And yeah, the position starts from 1, and then so 1 LPEA is the regular type, and then 1 GS9A chain A is the problematic ApoE4 type, right? And let me click this icon to make it the full screen. Right, I'm going to click to make it full screen. And then by comparing our previous result, it's somewhat here. Valine, arginine, glycine, VRG here. VRG around here. Let me come back here. And is there VRG somewhere here? Yeah, so I want to search for the 1GS9. There should be some VRG somewhere here. Yep, VRG is here. So the value is actually this around 111, actually 112. And then that is located here. Arginine is located here. But do you see this alpha helical chain? They almost match because the sequence is the same, except for the residue. In alpha helix, the residues are always standing outside from the backbone. So arginine should be protruding from here to this way. Arginine is the problematic one, and it's charged residue, positively charged one. But the cysteine is neutral, which is not charged. So let me just go back here and this is the arginine, and then do you see the difference in the residue here? Yep, this is the arginine and the cysteine. So what I'm saying is, yeah, question? I just clicked that here, 111, the number that I got previously. Okay. So what I wanted to show you is when you do the sequence alignment, yeah, you get, hey, this sequence is different. Amino acid, everything is same except for cysteine in ApoE3, and then the arginine in ApoE4. By doing this kind of structural alignment, you can actually see where it resides. And it's actually in this region. And if you actually these dotted lines are the hydrogen bond, and then the cysteine structure is actually is responsible. And this arginine structure, positively charged. Actually, alpha helix is the backbone. Our DNAs are mostly negatively charged, and that actually the arginine actually make a more stable alpha helical structure. And what is the role of A4B? A4B is combined with a lipid. But lipid binding region is in the C-terminus region of this protein. And then this problematic ApoE4 has a stable arginine, which is pretty fixed structure. So I will make it an open question. So think about how this arginine instead of a cysteine contribute to the function of ApoE4 protein, which is in the process of removing lipid in the brain. So the did you guys actually succeed this structural alignment? Yeah. So you initially start with the sequence level for the bioinformatics, but do not stop. Nowadays, we have a pretty good databases. You can easily go to the homology modeling, even have a modeling of your structure, and then get some insights from the structure. You can, if you have to structure, find the structure yourself. There is AlphaFold too, and those kind of software you can actually use. Yeah. All right. Why don't we take a 10-minute break? Yeah. And now the time is 7:14. Let's start at 7:25. Okay. So when there is a SNP, previously we were talking about one particular SNP in the that actually results in cysteine to arginine change in the amino acid. So let's actually try to understand the SNP more systematically.
is the SNP located in the protein coding region or not? For example, cystic fibrosis, the causative mutation is located in the coding region. So and obviously when we change the protein amino acid sequence, it may affect the function of that protein. So did you guys actually choose the SNP for your group 1? For group 1, did you choose the coding SNP in the coding region or not? You chose the coding one. How about group 2?
We're group 2.
Oh, group 2. Okay, group 1. Yeah, did you choose the coding SNP or the SNP in the coding region or not? Okay. How about group 3?
I think we finally realized it's
Okay.
but we are considering SNU after the course.
Yeah. Actually, you guys are more than welcome to change your mind whenever. But if you change your mind at the last moment,
so we change it.
yeah, and electronic SNP is fine if you know enough relevance to the gene regulation.
In paper, it says it's helped to regulate the exons afterward. So that's maybe that's how it works.
If you can clearly find out what's going on there, that's fine. All right.
But not
But if it's located in the non-coding regions, then you are actually limited, right? You cannot visualize. There is no change in the amino acid sequence. So you hope that is somehow regulated in the regulatory region, and then you want to prove indeed that actually do some impact on the regulation. And so I wouldn't say it's not impossible, but
I think the paper is clearly explains why it can help regulate the exons.
okay.
So so that's will make us hard to move on, or
Maybe that's fine. But did you find something that paper didn't explain? Basically, meaning that do you have your own group niche that the paper didn't address? Basically, your own hypothesis over to the if that paper did everything, then there is no reason to study further, right? So there should be something that the paper didn't do, so that you can think about your own hypothesis.
like so like for step 4, we are going to find another niche that is like some related with the one that's mentioned in the paper, or you will directly use step 1 to work with others.
So basically, your group decide that.
Oh.
Your group, yeah. So why don't you actually think about it?
Oh.
Yeah. I wouldn't say it's not impossible, but I'm going to say it's harder. Yeah. Yeah. And your group 3? I see. Okay. Yeah. How about group 4?
Coding.
Coding region. Okay, good. For the coding region, group 3 groups out of 4 groups are in the coding region. Do they change the amino acid sequence? We have a codon, right? 3 base pair change correspond to the 1 amino acid. So there is a possibility that even though DNA sequence changes, the actual amino acid sequence may or may not change. So that's what I'm asking. Does your coding sequence change? Does your SNP synonymous or non-synonymous? That's what I'm asking. Non-synonymous is the one that changes the amino acid sequence. So coding for non-coding. For the non-coding, I don't have anything to say, but for the coding, I can add. Does it change amino acid sequence? In that case, what do I call? Distance or the another alias for that is non-standard. That actually changes amino acid, and then synonymous the same amino acid, even the DNA changes. So I'm asking the three groups whose SNPs are in the coding region. Are you guys studying for non-synonymous SNP or synonymous SNP? Group one? Group one? Yes. Non-synonymous. This makes sense. Non-synonymous. Group three is non-coding region, right? No, that's fine. That's fine. Yeah. Group four? Non-synonymous. Good. And now I have one question. If some group study happen to be synonymous, is it worth it to study or not? If the variant is synonymous, would you continue to study or not?
Yes.
How many think people think yes? How many think people no? And why do you think it's yes? Yeah. Why do you think it's yes?
I would say for publishing it's fine, like more result is also a result.
Good spirit. But not the who says that it's not, I understand it with obvious. But let me make an argument for yes, you still need to study. When the DNA changes, even though protein sequence does not change, it may have some impact in DNA to pre-mRNA, matured mRNA, and then the protein sequence, right? In that stage, in the middle, there could be some association with the pre-mRNA and mRNA like formation, and there are some evidences, and also some epigenetic or enhancer-related association may happen. It's possible. So I wouldn't say don't study, but I have a limited resource. Then I would definitely start with coding and non-studies because that's like a low hanging fruit.
I have a question. So, what if you study the coding region but with a synonymous sequence? Will it still have any biological impact?
Yeah. So there are actually many papers on that case too.
Coding synonymous case. So association with the CTCF or some enhancer signaling or those kind of things. So if your SNP happen to be associated with that kind of feature, yes, that can be important, but chance is pretty low. So that's why if you have a quick resource and then you can allocate some alternative hypothesis it's fine. If you have that much research, yeah, why not? It's always good to check those kind of things. But if you have a limited resource and then if you have to try to shoot for the logging input, yeah, I will try. So it depends on not really the biology, but really the probability to succeed and then your research.
Okay. Okay. Now, SNPs can have some medical implications. Is there anyone who can explain the SNP that cause premature stop codon?
Or just explain it.
Yeah, the concept.
Okay, so if it's that means translation starts earlier than it should be, so it's a truncated protein, and in most cases it's loss of function, and it's not functional.
Yeah. So in that case, so the domain before that is fine, but domain after that is problematic. So it's really interesting. Protein is organized into domains. So if you have a long protein, some catalytic domain and some motif binding domain and things like that. So it actually the change to premature stop codon actually depends on if that happens here in the upstream, near the 5' end, yeah, this protein is not going to work. But if that happens here, and then this domain may not work. But if that domain is not really that important for the function, then it is still working. Okay. We will come back this example, RS-4, because I didn't introduce this RS yet. You have to know that what is RS stand for. Yeah, so let's just wait for 1 minute or a couple of minutes. And another example of the medical implication of a SNP is it may not cause the SNP, the disease itself, but associated with that. So LDL receptor reduces its efficiency and is associated with the risk of high cholesterol. And sometimes a SNP can be associated with differential response to drug. One SNP in the CYP2D8 gene disrupts the breakdown of a certain drug, which is used to treat the high blood pressure. Is this clear to you or some of yours? I'd like to go over this somewhat in detail, especially this pharmacogenomics. And this is actually a very interesting example. How many of you heard about pharmacogenomics? Did you actually have seen the real example of pharmacogenomics? No, you probably heard the concept, right? But let me introduce that. Genomics is a genomics based on your DNA, genotype, pharmacology, how the drug is responding to your genotype. That's the pharmacogenomics. You guys probably don't drink alcohol if you are taking the cold drug to treat your cold. Pardon? Yeah. Why you don't drink alcohol while you are taking some drug?
It increases the metabolism of the drug.
It,
The metabolic digestion of the drug will be enhanced and it will break down before it mentally. The action will not be delivered. Yeah.
action
Won't be delivered.
will not happen
Yes.
because
The metabolism of the drug has been proposed and has been to
Metabolism means
the breakdown of the drug.
breakdown of drug to the active form
Yes.
is increased or decreased. Yeah, it's both ways. Right. So the major reason why we don't drink alcohol while we are taking some drugs is to protect, mainly to protect our liver. In the liver, like many, the drug itself, most of the drug needs to be converted to another form to work. And that conversion usually happens in your liver by which gene? Cytochrome genes. Cytochrome genes are not the only ones that do the metabolism of your drug to change into the active form, but they are most popular. So you probably heard cytochrome P450 enzymes or genes, which are mostly expressed in your liver. So that's where that's happening. And if you take the alcohol needs to be converted to the aldehyde, and the aldehyde is toxic, and then it needs to be converted to water and the CO2, basically the H2O3. You basically give too much load, so it's not really desirable for the side effect, right? So if you carefully look at this, this CYP2D8 is one of such a cytochrome genes, which is responsible for breakdown of debrisuquin, which is one example of a drug. But some people have mutation in this enzyme. Then most likely this enzyme may not work so well. Then that metabolism is not so efficient. So what do you get if you give the normal amount of the drug to that person?
It will not be efficient.
Yeah, it's two problems. Not so efficient. Second, it could cause toxicity. So in that case, it's better to reduce the dosage, because doing no harm is the first principle. Yeah, curing is important, but cure as long as you don't do any harm. So that's kind of this pharmacoeconomics. Now, do you have understanding what's going on here? Okay. Now, let me just tell you some more. Now, so far, I talked about the standard material, but from now on, let me introduce the real industry here. So this is not really in the exam. This will not be tested, but I just want to actually share the current situation in the industry. Nowadays, you can get your DNA sequence, right? Whole genome, the market price is around $650 per person. If you use the stick chip, it's around $100 to $150. But the whole genome sequencing investigates 3 billion take the whole genome. The SNP chip usually check out about 1.4 million variants, predefined variants. Okay? 1.4 million is a lot, but compared to the 3 billion, it's a very small. So you know that the whole genome is definitely better. And accuracy, I mentioned last week, SNP is the 1% error rate, roughly speaking. But the NGS, the whole genome, even though the machines, the error rate is around 21%, since you pile up and get the consensus of it, when you do the variant calling, the error rate goes down to 10 to the minus 5 or 10 to the minus 7. So it's obviously better to use the NGS. Okay. After you get the doing the NGS, can you
get the genotype of cytochrome gene? Not just this cytochrome gene, but all the cytochrome genes in one shot. Yeah, why not, right? And then, can you check out the genotype of that, the cytochrome genes? And can you actually provide that to the physicians who is trying to prescribe the drug so that they can be proactive or do the personalized practice? So that's the personalized medicine. In theory, everything is good, right? Where are we now? In reality, in the industry. So if you pay about $700, you will get raw data, FASTQ file. You'll see that FASTQ file soon, like in a month. And then you process a line to get the BEM file, variant calling to get the PCF. And now you have 3 million variants in one big PCF file. And when you use some software, you get genotypes of all the cytochrome genes. Okay. And as of now, some companies like the Genome use the SNP technology to give you the report on mental disease related the report on this pharmacogenomics, which is about $300 in the marketplace. And there are many like companies or hospitals that provide that. And why really need this kind of technology? It's better than nothing, right? You can avoid the side effect and you can protect your liver. So it's better. But when this really becomes important, this pharmacogenomics. Medicine is expensive. Yeah, that's good. Yeah, that's another good application. But most helpful case that I think is when someone just gets diagnosed as a cancer. How many of you heard about the chemotherapy? Most people know that, right? And do you have to know that you have only one chance for the chemotherapy? Why? Because of resistance. Did you hear about the resistance? Resistance of a cancer drug is different from. So I'm not afraid of the resistance from the normal drug, but I'm afraid of the resistance of the cancer drug. Why? Because in the cancer case, it's called multi-drug resistance. When you develop one resistance, usually we develop for that drug. If you avoid that drug, you're fine. But in the case of a cancer case, it's not.
in the cancer case, if you develop a resistance for drug A, you simultaneously develop a resistance for BCD or for almost all sorts of that kind, the steroid type. So if you do not get the right drug in the first trial, your chance to survive is pretty low. So that's another very significant pharmacokinetic. Just to since you guys are going to in the science field, you might wonder why this multi-drug resistance is happening. Usually, if you give some drug A, you should develop the resistance for the drug A. But why not? Why for all the drugs? That's because of this kind of multi-drug resistance technique. So I happened to be for my first postdoc, I was in the NIH, NCI, the Cancer Institute, and that lab happened to be studying this multi-drug resistance. And when you have a cell, you have a membrane protein. There is a membrane protein that transports the drug molecule into it. For the cancer drug, it's very toxic, and when you give a drug A, and then if it cures perfectly, then you are surviving this. But if you keep giving this drug, cancer like a chemo drug, this what is that? This membrane protein no longer work as a channel protein to transport it into the cell. The pumping out of it without getting in, this is actually activated, and that's universal for not just one type of a steroid, but all sorts of a steroid steroid. So that's why when you do the chemotherapy, be careful for which drug you choose. You want to choose the best drug. But nowadays, situation got somewhat better because we have nowadays some new types of a drug. Previously, we just have like a steroid type of a drug, but now we have an antibody drug and those things. So that's kind of much better use. But anyway, pharmacogenomics actually, but antibody drug too. Antibody drug binds to the receptor, right? If you happen to have the severe mutation in the receptor, then the drug will not work properly. So for the cancer, the pharmacogenomics, I think that will get more and more attention in the future. Yeah. Yeah, there are actually some, and yeah, so I was somewhat right? Yeah, let me come back. So the studying SNPs are important because not just for the relevance to the disease, but also you can do the personalized drug recommendation to avoid the side effect and to find the drug that might work better. And this is not just reducing the side effect. For the case of a cancer patient, this may life-saving like the test. Any questions so far? Okay.
Now, the categories of SNPs, we already searched it, right? The coding region protein versus a non-coding region, and the coding region SNPs, the synonymous or non-synonymous. And in the example of this, that ApoE4 SNP, which changes cysteine to arginine, that is belong to coding region C and non-synonymous. Is it clear? Now, how many of you have experience of searching DBSNP? So DBSNP is the database of the SNP. It may contain the SNP, right? But not just the SNP. It may contain the insertion and deletion too. So even though the name says a SNP, now it became somewhat generalized. So database of variants might be better name. But anyway, they are just keeping the DBSNP. So everybody, can you actually try to search this RS429358 in the DBSNP search box? Because whatever it starts with the RS, that is the ID of DBSNP. So when you register a SNP in the DBSNP, they assign RS ID. RS stands for the reference SNP. So from now on, if you happen to see RSID, okay, this is the SNP registered in the DBSNP database. That's what I wanted you to know. And did you find the this particular SNP? Can we take a look at that? This RSID from this database. RS429358. So this is the DBSNP. It contains mostly the short genetic variations, SNPs and short insertion and deletions. And this is the for Homo sapiens. According to the Human Genome Research Consortium's reference genome for human, version 38, major version 38, minor version 14, chromosome 19, and at this position, reference allele is T, but the SNP allele is C. Now, I have one question before moving on. It does not tell you whether it's a 5' to 3', the forward direction or reverse direction. Why it doesn't tell you? Because everything is No. If it's forward direction, it's T. It's reverse direction, it should be A. Right? Exactly. By default, even though gene is located in the reverse direction, in the DB-SNP database, they always change it to forward direction. So BRCA1, the direction of the gene is actually located in the reverse direction. But if you actually look up the DB-SNP data, every sequence is translated into the reverse strand, the forward strand. So for those genes that belong into the reverse direction, you should be careful because this actually doesn't match. You have to change A to T like a complementary base. Okay, and since T is going to change into C, replaced, this is single nucleotide variation, SNV. And which one is major allele? Which one is minor allele? Usually, major allele is the first one. Minor allele is the SNP case, right? Usual case. What is the allele frequency of C compared to T? Out of 690 cases, 41,110 cases has C at this position. So allele frequency is around 15%. Right? According to TopMed database. Though they are the aggregated database of the genomic data. And depending on which population you sequence, allele frequency changes slightly. So in the Genome exome sequencing project, this allele frequency is not 15%, it's just 13.8%. But the regular Genome database, allele frequency is higher. But when I actually look at this, the average is around 14 to 15%, right? So, okay, 14 to 15% plus minus 2%. That's what I actually read when I look at this. But here's a catch. This allele frequency is specific to each the ethnicity. So European American versus Korean versus those are very different. Asian versus so in which database you basically if you go to this database, you can actually check allele frequency for each ethnicity. Yep. So if you click like more here, the Estonian people, 12%. Korean people, only about 9%. Like that. So it's ethnicity dependent somewhat. And clinical significance, is this SNP relevant in the does this SNP is relevant to the disease? Go to another database called the ClinVar. Okay, let's go there. So DB-SNP is not really the database that tells, okay, if you have this SNP, you will get this disease. No, DB-SNP is just a database of a SNP and let you know rough the allele frequency per each ethnicity, assuming that SNP is in the forward direction. And this, it's an ApoE gene and then missense variant, meaning that protein coding region changes amino acid. this SNP is studied extensively, already 484 citations. This is exactly what I want you to avoid, because if you have a couple of hundred publications, it's not easy to think of your unique niche. Without the niche, it's not easy to publish. So if you have that much fully studied, it's better to avoid instead for the purpose of getting the low-hanging fruit. So that's why I keep telling you, why don't you try for the novel SNP? Novel SNP doesn't have to be really novel. Nobody studied, but I hope that the number of publications are less than probably 10 or 20 or less than 50. And yeah, so here is some more detail of allele frequency in each population, African, Asian, like that. And this number is somewhat different from this number, right? Hey, Korean, 9%. But why this, if I come here, Asian, is the Korean is the 9%, but C is only 3%. East Asian, like 2%. What's going on? Yeah, it actually, everything depends on the target population. Target population is not exactly the same. For some, they recruited the people from Korea. For some, they recruited the people who claims Korean in US. And that cannot be the same, right? So, so allele frequency, what I'm saying is, it's very useful information, but don't trust too much because there is a huge variability between the databases. Just be aware of that. So yeah, and then here is the variation viewer. The reference genome sequence is T. In the forward strand, in the backward strand, it's A, right? They show that not only the forward strand sequence, but also the backward strand sequence. But the SNP sequence is C. So T becomes a C, and T becomes a C in the fourth strand. If it's in the backward strand, C becomes a G, right? So it's a T2 C the mutation or A to G mutation in the backward strand. For the case of BRCA1, which is located in the backward strand, you have to interpret the other way. You first convert this T into the A. Reference sequence of the mRNA is A. And then your SNP allele is not C, but G. So A to G, like a mutation in mRNA sequence. So it's complicated, but you have to do that kind of step. Otherwise, the result won't make sense. And do you see that the RS429.358 T2C like variant is right there? From the DB-SNP database, version 1.56, version 2, and there are multiple like this kind of mutation like a viewer. So this is the DB-SNP database. Do you have any question? It's pretty simple database of, hey, this is the T2C SNP in the chromosome this position. If you want to know some more like association to the disease, try to go to another database. That's what it says. Yep.
So like the table we found is February 2022, so it means that we don't have even updates in previous.
Okay, this following information is last updated on this date, but this DB-SNP was there for about 10 years. They updated the part of this information.
Yeah, this is when we are like checking our research, we try to find the information on database, but we can check there is some information of the new code. So we are wondering if that is because the database still will update till the paper is published or something. Is that possible or?
Yeah, actually, two things are happening simultaneously. What you said is actually happening. So in that case, you actually go to this PubMed database, Genome database, and you actually go and there and check it.
To see which is the latest.
Yeah. Or allele frequency, if you want to check the allele frequency. But the second problem for your group is the non-coding, right? Most of these studies are exome sequencing projects, so it's not there. And even though it's there, it's limited. Yep. So yeah, those are actually one difficulty that you already face. Yeah. Any other questions for the DB-SNP? So now you know that each SNP or variant is registered in the DB-SNP database, and that has the RSID. And the RSID, you have the position close to 19 at this position, T2C6, right? If you are interested in the clinical significance, go to the ClinVar. When I click that, this is a ClinVar. This SNP is in the gene of ApoE, and then there are many pathogenic like association reported. Yep. But ironically, even there is the protective thing. So be careful. You have to actually read the paper. And do you see this NM? If you actually click that, oh, here, let's check the this ApoE. The T2C SNP at the position 388, and this particular one is associated also with the familiar hypercholesterolemia too. Not only that, the Alzheimer's, like this paper, there are multiple citations. Now, I want to explain something. For a given SNP, I think that nowadays, so many people are abused with weather focus. So what is my probability that I'm going to get the Alzheimer's disease if I have ApoE4 mutation? That's one factor. And which population you study is another factor, right? And that population has a multiple ethnicity group. That's another factor. And you have not just this factor, but Alzheimer's is caused by not just one gene, but multiple variants, right? That's another factor. Do we know everything about Alzheimer's? No.
then how come some companies or someone actually predict, hey, your chance of getting the Alzheimer's is 72%. Pardon?
Yes.
Yeah. So this is my personal view. Honestly, I don't believe all these kind of percentages because now you know all these complications, how much we know it, and then if you look at this evidence, do you see that the publications and some publications actually collide? In this case, it's pretty obvious, but in many cases, some say, hey, this mutation caused this disease. Some say, no, this has a protective role. So they have a different opinion too. And investigation, how actually can you predict the likelihood of like getting that disease? Because the data that we have, I just introduced all the data that we have. Only frequency for each group, and then some list of publications relevant to that. And then list of publications is based on their own small population or in vitro experiment, which does not account for picture of Alzheimer's disease. So it's just a hand waving guess. So that's why I think that the approach of predicting percent of the likelihood of getting that disease, because that particular SNP is most likely wrong. Okay.
Right? So I think I fully introduced the DBSNP, which is very important database. Yeah, again, the RSID, RS stands for the reference SNP. But the SNP related database, is there only the DBSNP, or are there any other databases relevant to the SNP? Here you are. Barsom, SNP Effect, SNPpedia. Can you actually try to search this RS.410.635.01.FA23 the one that you just searched in the DBSNP in each database? And then your group is going to study on the role of your SNP. And which database would you prefer to get started with? Yeah, why don't you try now? Yeah, if you guys want to talk to each other in your own group, yeah, feel free to talk to each other. Because yeah, you'd better do the good communication. And after you search all these three databases, I would like to give some warning of like a caveat of certain databases, such as SNPpedia. Yeah, let's get started about in 3 minutes or 5 minutes so that you guys can discuss in your own group.
Can I ask one question for you? I sent the exit candidate.
Oh, yeah.
And it's fire as
That's fine. Second part. Wait, racing. Oh, that's what. They actually did.
Yeah, the trick was I was using the company email,
Some of the questions.
but yeah, pardon?
So we have dots on them. Like dots. Dots.
Oh, yeah.
Like the name and the red dot.
Red dot?
Yeah.
What is the red dot?
Right here. There's no like there's no there's no this says
Just delete.
Uh,
Interesting. uh, because what I'm actually looking at is this. This is a Unit 6 version 2. Yeah. Send, share link, copy, and then the chat. And this is what I did. Pardon?
The second and third questions I'm talking about. I mean, if you just pull that out, we can talk to this.
Pardon?
You just have that. Yeah, we can just move that.
Oh.
Exactly. You guys have a question.
Oh. Let's just delete that. Delete this.
And there's one more.
Oh.
Yeah.
Okay. Yeah. Yeah. Okay. Let's continue to the. Yeah, I'm very glad that finally I found the solution for exit ticketing. The trick was use a regular Gmail, not really company-based Gmail. And there is a big difference, even though those are using the same Gmail platform. Company Gmail is somewhat like a Google Form is only working for company members, I guess. Yeah, that's what I think, the current hypothesis. All right. So how many of you prefer the Barsum? Please raise your hand. How many of you prefer the SNP effect? How many of you prefer the SNPpedia? Okay. Let me just give you one warning for the people who prefer the SNPpedia. It's notorious knowing of containing SNP information which are wrong. Yeah, I wouldn't say yeah, but it got better over the time. Just like a Wikipedia, anybody can come and then edit it. Pardon? Yeah, it's a concept is the same. SNPpedia is basically Wikipedia. Yeah, so but the Barsum has some kind of a filtering. So my personal preference is the Barsum, but over to the SNPpedia. But sometimes Barsum doesn't contain all the information. Sometimes the SNPpedia has that. In that case, I have to look up, and it's better than nothing, right? Then I have to. For the SNPpedia information, yeah, I can be more critical in evaluating. And so the same is true for the ChatGPT. A lot of the ChatGPT is trained on the Wikipedia and then the Reddit, right? So yeah, so just everyone by now know that what the ChatGPT says is not 100% true, right? Yeah, think in the same. In my mind, I think the same like way for the SNPpedia. And unfortunately, the same is true for DBSNP. DBSNP, unless it says expert curated, yeah, be careful. All right. So now ClinVar. ClinVar is, DBSNP contains a database of a SNP, and relevance of that SNP to the clinical phenotype, they simply say, hey, look up the ClinVar. So you can search the ClinVar by gene name, like a CFTR square bracket gene. Can you actually try it? Can you go to the ClinVar and search this CFTR ClinVar? right? Let's do it together. ClinVar and CBI, and search this CFTR by gene name. There are many mutations, pathogenic mutations, likely pathogenic mutations, uncertain significance, likely benign, conflicting evidence, mutation with a conflicting evidence, not provided other. So, and then do you see that the GRC H37, which is not H38? So be careful. And if you just click it, it will change into the HG38 version. And this gene is the like a big gene, so it contains many variations. And per each variation in that gene, pathogenic mutations and uncertain significance like that. So if I just click this cystic fibrosis, let's try to find the pathogenic mutation with cystic fibrosis. Okay, pathogenic. Let's look up. So this is interpretation of in the ClinVar is a pathogenic mutation, and VVVAR link and even varsome link. So let's click the varsome link. Yeah, this mutation is too big to visualize. Let's look up the some simpler case. Yes, this one. Uncertain significance. Just the one submeter provided that. And then condition is relevant to the cystic fibrosis. And there is one publication linked for that. And DB-SNP RSID for that mutation is here, RSID. And you can have a varsome link too. And in the cDNA level, 837's the DNA sequence is T, but here it's a C, T2C mutation. This is the earlier ID in the ClinVar. It's a SNP, one base pair case. It's located the Q arm of chromosome 7, and the band is 31.2 cytogenetic location. Genomic position is chromosome 7 at this position according to GRC8038. If you want to go to the genome browser, you can click those. And some other databases like HGVS or like some other links too. Unfortunately, the early frequency is not here, so you have to actually go to each GenomeAd or some TopMed database and actually search the RSID. to get the idea. So basically, in a nutshell, the take-home message is, if you want to check out clinical association of that SNP to the disease, start with the ClinVar. That's the simple message. You can either search by gene name, then you will have a lot of variant associated with that gene, or you can actually search by RSID. Then you get that particular one in the ClinVar. Any question? And ClinVar data is also available in the Ensemble, UCSC, and the NCBI. So you can visualize together with those genome browser. Okay. Now, the copy number variation. What is the copy number variation? You probably heard a single copy, multiple copies of the gene. For example, some gene 1, can be located in the multiple locations of a chromosome. Even in one chromosome, multiple positions, the same copies. And in some case, everything can be the protein coding. In some case, it's not. Pseudogene, meaning that, it can produce up to the RNA, but no protein coding. So all these cases are happening. So how to detect the copy number? Let me actually show you the two different approaches, which is not actually written here. Two popular technologies that check the copy number variation is one is by chip, the chip. The other one is NGS. So I'm going to introduce both of them. The chip is significant compared to this. It's called a tiling array. So for example, for the first block, chromosome 1, first position to 1,000 position, or chromosome 1, 1,001 to 2,000. So you just tile this. You have about 1.4 billion the rooms here. And then you break your DNA, and then let it hybridize it. If it's just in a certain kilobase or certain megabase, if there is a copy number variation, that may have double copies or triple copies. Then the average intensity is, let's say, 1.7. Average intensity is like that. But in some regions, it's going to 3.4 instead of 1.7. Then, okay, this region, they have like a double copy, depending on that intensity, the degree of hybridization. That's a tiling array technology. But NGS is different. In NGS, we just align, and then here is the chromosome 1, and then first base pair position, and chromosome 1 is all the way to the 250 million base pair position.
And you just check the what is that? How many reads that in that video, and draw the histogram. If there is the copy number variation in certain region, it's like that. So tiling array, you basically make a tile and then tile is prefabricated, and then if you see that higher intensity, yeah, that region is the with more copies. In NGS, you just align it and then check out the histogram. And the morphology-mediated break-induced replication may explain why the copy number variation is happening. The mechanism of that MMBIR is it starts initiation when the double-strand break DSB occurs or replication fork collapses. The DNA ends undergo the resection to expose the single-stranded DNA. Single strand is SS. Then microhomology search, this tail part of the ssDNA search for this microhomologous sequence, usually around the 20 base pair elsewhere in the genome, and that the strand invasion follows because that ssDNA, single-stranded DNA invades the donor DNA strand displacing the original strand, basically interacting. And then replication start, and then finally followed by the termination. So that's the simple mechanism of why the copy number variation can happen biologically, according to that paper. But I would like you guys move on one more step further. We started with the SNP. SNP is replace of the base. Copy number is you have another copy inserted. What is the general case? In general, we call structure variant, SP. So structure variants include the copy number variant that we just talked, but that's not limited to that. It has gene deletion. The chunk of a gene can be deleted. And which can be like another sort of a copy number variation, right? Gene can be duplicated. Yeah, it's another type of the copy number variation. Some part can be inserted, deleted, inverted, put into another location, translocation. All these things can happen. And multiple regions can in a complicated way, mix and match and combine. Where these kind of things that happen most in reality? In the tissue of cancer. Tumor, yeah. If you dissect the tumor and sequence it, you will see all these kind of things that happen. And traditionally, we define that like a chunk, like at least to be eligible for the structure variant, it should be like 1 kilobase or larger. But nowadays, yeah, if it's over like a 50 base or 100 bases or 70 bases per long, then yeah, many people consider that should be classified as the structure variant. So, My final message for today is we started with a SNP, single nucleotide replacement in the genome. And we introduced, I'm just wrapping up and then you to understand the take-home messages. The databases that has a lot of SNP-related information is that you want to start with a DB-SNP. In that DB-SNP, each SNP is registered as a RSID, reference SNP ID. But it's not limited to that. You can search the Barform and Wikipedia, all these, but be careful about the information. Databases are just the aggregation of information. You have the responsibility to check whether that information is trustworthy or not. And especially for your group project, you are studying one SNP, not the hundreds of SNP or thousands of SNP. You are studying just one SNP, right? In that case, what I recommend is start with those SNP-related databases, but don't rely on that information in the database. Go to the citation and go to the exact paper that's referring to, read about those references, and try to understand your SNP, not just from the summary statement of the database, but try to understand it from the paper, original papers. Why? I saw a lot of mistakes from paper to the database summary. If you are studying for thousands of SNPs simultaneously, yeah, I understand. You may not want to check all these details one by one, but you guys are studying for one SNP. Then I want you guys spend, if you go to DB-SNP, there are 10 or 100 references relevant to it, right? And for each SNP paper, they cite 20 or 30 or 70 papers. So you have a lot of original source. So my take-home message is just don't rely on what's written in the database. Always try to have a habit of going to the original source and then validate or verify to minimize potential the error. Yeah, because I have seen so many such cases. That's one take-home message of today. And another take-home message is DNA sequence alignment, the protein sequence alignment, and the structural alignment. So if your SNP is happen to change the amino acid sequence, then try to visualize the 3D structure and try to understand from structural view also, not just the sequence, because sometimes structural understanding helps you to understand better. Yeah, those are the basically two take-home messages. And if you have time, yeah, please fill out this exit ticket. Yeah, and if you have any questions, I'm going to stay here, so you can ask me. Yep, thank you.